Okay, so we're recording again now, back from the groups. So as I said, I hope that you were able to determine which group you were in and what you were meant to be focusing on. So the first group was the group that had Bassem and Kiani in it. So you guys were to list the factors that influence the AHG test and explain how they do so. So would somebody in that group like to let me know what sorts of things you came up with, either on the chat or in the or just unmuting your microphone.
We came up with temperature. Usually, I suppose, when we heat things, they go faster. We came up with human error, so just misinterpreting things or just putting the wrong things in, not following methods. A ratio of serum to cells. Jeff talked about something to do with competition this morning, so I don't know if that's the right idea, but it sounded good. And the reaction medium.
So when you're talking about reaction medium, what sorts of things do you think?
Well, again, didn't quite understand it, but this bit that I was reading was talking about the macromolecules of albumin being adsorbed onto the red cell surface and helping aggregation occur.
Wow, excellent, great. So anybody else from that group come up with anything else?
I just stole their ideas. They come up with their ideas with me.
So that's really great. Well done. So we'll go on to the next slide, which actually, so these are a list of factors affecting the AHG test, which are listed in Harmony. So as you can see, you have included a number of those. So you talked about the cell to serum ratio. So why do you think this might be important? The cells and the serum or the plasma. Why do you think that might be important? Competition, yep, too little cells, can't see agglutination well. So yeah, so you need enough cells so that you've got your antibody there, and you need enough, you know, that you've got antibody on your red cells there, so enough sensitized red cells for the anti-human globulin reagent to actually react to. And so to do that, particularly with your indirect antiglobulin test, of course, this is more to do with, so you need enough antibody in your plasma to react with the antigen on your cells, and then that sensitizes your cells, and then you've got enough sensitized cells to react with the anti-human globulin. The reaction medium, yes, that is very important because it's actually the reaction medium that helps to reduce the zeta potential. So you heard that Kiani mentioned albumin. So albumin is one thing that can actually coat the red cells and reduce that electrical charge and therefore reduce the electronegativity, allowing the cells to get closer together. And we actually do use a different reaction medium in our anti-human globulin test, which is called low ionic strength saline. And that also reduces the electronegativity of the cells and allows them to get closer together. Temperature is really, really important because particularly when you're talking about IgG antibodies, they react best at 37 because that is human body temperature. If the temperature is too low, then the antibodies will not adhere, and if the reaction temperature is too high, that can be denatured, so that's very important. Again, incubation time, you have to allow time for the antibody and the antigens to interact and to bind. Washing of red cells, I don't know if you talked about washing of red cells, but again, this is to make sure that we remove any of the free antibody and plasma that is there, so that all we have left are the sensitized red cells. The saline, so that's important that it is buffered normal saline. So it is of the correct pH because pH can affect antibody-antigen interactions. Adding the AHG is extremely important. If you don't add the AHG, then you're not going to get the bridging between your sensitized red cells and you're not going to get the agglutination. That's why when you have a look on the video or when you go into the lab, you'll notice that the AHG reagent, the Coombs reagent, is coloured green. So it's really, really easy to see when you've added it and when you haven't. Centrifugation is really important. The time of centrifugation, the force of centrifugation. So you don't want the force too great so that you're going to get pseudo-agglutination. You know, you're just going to ram those cells together, and so they're going to look like they've agglutinated when in fact they haven't. And you don't want it to be too light because you want those cells to be pushed together at just the right speed. Okay, so that's why you've got two settings on your immufuses. You've got the longer, harder setting for when you're actually washing your cells to get rid of the extra plasma and the free antibody, and then you've got a shorter, lighter spin for when you're actually wanting to help with the agglutination of your cells before you read them. Okay, that's why you've got the two there. Well done, people. That was great, particularly as we haven't really been talking about these factors before. Okay, so group 2, you were asked to think about factors that would affect or give us false positive errors. So what sorts of things did you come up with? So this was the group that Amelia was in. If somebody would like to unmute, it's probably a bit faster to unmute.
Yep. I think what we talked about was actually more of not properly washing the red cells. That's the first thing, I think.
Mm.
And then we talked about having using an improper sample, such as not using the right tube of blood for the IAT.
Mm-hmm.
And as well as spontaneous agglutination.
Mm-hmm.
And certain diseases which cause a lot more antibodies, for example, nephritis, or maybe some kind of cancers, perhaps, and some non-specific agglutination that may be genetic.
Oh, okay. Well done. Excellent. Really, really good. I noticed that, yes, you had different specimens and things as well. Yep, that's great. So remember that whenever we're talking about factors affecting reagents or sources of error, it's talking about human error is really important. And I think you did that when you talked about the improper specimen. So that's really good. So make sure you've got the correct specimen. So when I was in the lab, we actually used to use, we weren't all that fussy about the samples that we used. And I think we used to use clotted blood samples. These days they tend to prefer to use EDTA samples because it is known that when in the coagulation process, you can actually activate complement within that process, and so you can end up with a false positive through that. And so that's one of the reasons why they like to use plasma from an EDTA specimen. Also, you know, if you get an incorrect, you know, you mix up your specimen, so you might, you know, again, mixing up your cells in your plasma or using the wrong one. So you talked about polyagglutinable cells and autoagglutinable cells. So again, we learned a little bit about that with our ABO discrepancies. So some of the same things apply. And if you use cells that have already got a positive DAT, then they will be positive, you know. So that will give you what would be considered to be a false positive. Any contamination may cause the cells to agglutinate or may give you a false positive as well. Over-centrifugation, so once again, if you're using the wrong speed for the wrong time, and also as you get better at reading your reactions, you'll find that some people are a little bit too hesitant with how they tilt their tubes and how they look at their tubes. It's really important to look at how the cells flow off the button at the bottom of the tube rather than actually looking at what's happening in the tube itself. And if you go back and have a look at those reading the strengths of reactions, then I think Sam mentions it in that as well. And certainly Melina will be mentioning it in the prac this week. We can get what we call antibodies that will interact with some of the reagents that we're using. So one of our very rare group discrepancies is that we actually have antibodies that will react with the, not antibodies, there are some antigens on surface that will react with the anti-B, anti-sera in the ABO blood grouping. And similarly, we can get, it's very, very rare, but you can actually get preservative-dependent antibodies using our low ionic strength solution. So there are a number of sources of error for false positives. Okay, so for our final group, what about error for the false negatives? What did you guys come up with? And this is Adelaine's group. Again, if you can unmute your microphone, it's going to be faster, I think.
Well, we had a list that we got from a website, but I brilliantly forgot to copy that. But it's fine, I'll just refer to the textbook. So I guess first of all, we have improper washing of cells.
Lovely.
Yeah, and of course we could also have poor technique, so like you don't actually know how to read and interpret the results, or like you forgot to add the reagents itself.
Lovely, yes.
Yeah, and also it's possible that you have false negatives because of non-reactivity, which would be associated with maybe the reagent has expired, or maybe the serum itself is non-reactive because of the lack of biologically active complement. And you also, it could be because that the cell suspension is too weak or too heavy, and like related to that also, false negatives could occur with under centrifugation or over centrifugation.
Lovely, well done. I think you've covered absolutely everything. Well done, people. So yeah, so this is probably inadequate or improper washing of cells. So it could be that you haven't washed the cells well enough and there's still some free antibody there, or it could be that you've overwashed the cells and you've lost a lot of your cells. So that would be that would affect your cell to serum ratio, wouldn't it, or your cell to plasma ratio. So it's very important to use the proper cell washing technique so that you wash your cells adequately, but you don't lose too many of your cells. This is a big one, AHG reagent not added, but of course, as I said, that's why it's coloured green, so that makes it very clear to see whether or not you've added it. Forgetting to add the plasma is a huge one, and of course, because that's not, you know, it's just normal plasma color, and it has to be added to your indirect antiglobulin test right at the beginning. So when you're setting up your test in the first place, make sure you've added your plasma. That's really, really important. Yes, you talked about most of the things that are in this list. So remember, you have to make sure that you have correct incubation conditions. Really, really important to make sure that your water bath is at 37 degrees plus or minus about 0.5, and that you incubate for the correct time. And then, yeah, so we talked a little bit about that. And again, proper centrifugation. And as you get better with your reading technique, you will get better at this, because particularly with the AHG reactions with some of our other blood groups, it's very easy to actually shake away reactions. So you have to be very careful there. Well done, Group 3. That's excellent. And just to show you, that's the AHG reagent that we use, and you can see it's quite green, and it's also got a green lid on it. Okay, so we better move along fairly quickly now. I think we've still got quite a bit to cover. So what do you think in this? Which factor affects the antiglobulin test but is out of our control? Antibody affinity, gravitational force, incubation time, or temperature? And I'll just let you have a think about this rather than writing up the alternatives, because we're running out of time a little bit. So is it going to be A, B, C, or D? Beautiful. Well done, Kiani. Yes, it is in fact A, the antibody affinity, because gravitational force, that's the speed of our, or the force of our centrifuge, we can control that. We can control the incubation time, we can control the incubation temperature, but we cannot control the antibody affinity. That is intrinsic to the antibody itself. That depends on the blood group and the class of antibody that it is. Some have higher affinity than others. Well done. Okay, so now we're going to go on and have a look at how we produce our AHG reagents. And as I said before, we usually use another mammal, and you can see here that rabbits are used. And depending on what we're trying to achieve has an effect on how we actually make our reagents. And as I said before, very broadly, we put a foreign protein into non-human mammals. That foreign protein is our human IgG or C3D, and this induces an immune response. We then bleed those animals, process and isolate the anti-human globulins. So you can see that we can use either IgG or complement components, and it's usually C3D that we use when we're looking at our complement components. Okay, so there are different types of reagents used in the transfusion laboratory. So you can have polyclonal, monoclonal, polyspecific, and monospecific reagents. So when we're talking about the clonality of a reagent, that is the origin, so that's how it's made. When we're talking about specificity, this is the target, whether we want it to be specific for a certain target, for a certain component, or whether we want it to be more general. Okay, and each of these has advantages and disadvantages. So we'll have a look at that now. And so first of all, we'll look at polyclonal versus monoclonal. So polyclonal is usually prepared in rabbits, and as you can see here, it is pulled from a large number of donors. So that's where the poly comes in. It's not just from one animal, it's from a large number. And so you also get serum from a pool of human donors. You isolate the IgG fraction and the complement fraction. You inject the IgG fraction into one colony animals of the same species, and the complement fraction into another colony of animals of the same species. You bleed the animals, collect the serum, so you end up with polyclonal antibodies. So the antibodies are produced by a number of different plasma cells from a number of different animals. This is how most reagents are made, although because it's a lot cheaper to do it this way. Although these days monoclonal techniques with the hybridoma techniques are really bringing down the price of the monoclonal reagents. So typically mice are injected in monoclonal reagents with a purified human globulin or human complement. Then the spleen B lymphocytes are collected from the mice, and they are fused with a myeloma tumour cell line, because we know that myelomas produce lots of immunoglobulin, don't we? And they produce a hybrid, that's the hybridoma. These hybridomas produce monoclonal antibodies, so their antibodies are exactly the same. And then these are produced on a mass scale in fermentation chambers. So the monoclonal antibodies are now becoming a lot cheaper than they were. Okay, so this has the advantage of being highly specific in how it's been made, whereas this one is less specific, so it's more general in its application, I guess. So this is basically what we've been talking about. So it's summarising how the different reagents are produced. And so if we do this first, so this is our polyclonal anti-human globulin. So as I said, we usually have two groups of rabbits, one that we inject with IgG, one that we inject with complement. And then we mix the product together, and this produces a polyspecific reagent because we're mixing some anti-complement antibody and some anti-IgG antibody together. So that gives us our polyspecific reagent. So it's specific for more than one thing. And it's made from a polyclonal blend because we're using more than one animal. Okay, does that make sense to everybody? So polyclonal is where it comes from, the clonality, where it comes from. Polyspecific means, or the specificity means what it's going to react with. So a polyspecific reagent is going to react with complement components as well as IgG. And you can see down here that if we just purify the anti-C3b and C3d by itself, we get a monospecific anti-complement reagent. And if we do the same with our anti-IgG, we can get a monospecific anti-IgG reagent. Okay, so this process we can get monospecific reagents and a polyspecific reagent. Does that make sense? The monospecific anti-IgG reagent is in a box because that's actually the one that we use in our laboratory. And the reason that we do that is because the samples that we use to make our patients for you have all been frozen, and they've been frozen for a long time. And so we know that that freezing and thawing process can actually activate complement. And so that is why we don't use a reagent that's got a complement component to it, because we don't want you getting those false positives, which will come from that freeze-thaw process. Okay, so that's the one we use in the labs at uni. So when we're looking at hybridoma technology, I can see someone's just asked me a question. So polyclonal means more than one rabbit donor. Yeah, no, more than one rabbit donor. Okay. So when we look at the hybridoma technology, yep, so usually it's rabbits that are used for the polyclonal reagents. When we're looking at the hybridoma monoclonal technology, we tend to use mice, and we would use one mouse. And we would use the B lymphocytes from the spleen of one mouse, because that's all we need. Because once we bind it with the myeloma cells and create our hybridoma, then that will just go ballistic, and that will produce exactly the same antibody all the time. Okay, so that's the difference between the two. The monoclonal ones are much more specific and less likely to cross-react. Okay, so we have one mouse that's injected with human complement, and then, as I said, the spleen is harvested, and then the B lymphocytes are bound to myeloma cells, and we get our monospecific reagent that way, or we inject IgG, human IgG, into a mouse and again harvest the spleen, do the same thing. So we can get our monoclonal monospecific reagents that way, and we can also have a monoclonal polyspecific reagent just by mixing these two reagents or these two products together. Okay. It can be very confusing. Sometimes it does take a little bit of time to get a handle on how that works. So just remember, polyspecific is a blended reagent of anti-IgG and anti-C3 antibodies, so it will detect both IgG and C3, and we use that as a screening reagent. Monospecific is when we've got a reagent that is specific for IgG or a reagent that's specific for C3, usually C3d. Okay, and so the one that we use in the laboratory is a polyclonal monospecific IgG. Okay, so remember that clonal means the origin or the derivation, and specificity means the target. So if you keep that in mind all the time, then that should help you. And again, that's just a summary of everything that we've been talking about there. I'm just cognizant of the time, so I think we need to move on unless anybody has got any questions about that. Okay. And this is just another way of summarizing it. So reagent, polyspecific, anti-human globulin, anti-IgG, anti-C3d. Monospecific is anti-IgG only, monospecific anti-C3d only. Okay. So again, this is just pointing out this C3d, which we're picking up rather than looking for, some of them do have C3b in them, but these days it's more C3d. And as I said before, that's because the C3d tends to hang around on the cell membrane of when there is IgM activation. And so it's the C3d. If we find that there, that's an indication to us that we've got a cold autoagglutinin, or we've got some sort of intravascular hemolysis due to IgM. And so the final part of today is looking at the clinical significance of the direct antiglobulin test and the application of it. So when do we use a direct antiglobulin test and when do we use an indirect antiglobulin test? Okay, so the indirect antiglobulin test is when we're trying to check our patient to see if they have got any unexpected antibody in their plasma that might react with donor blood cells and cause intravascular hemolysis or extravascular hemolysis in the patient. So they're not going to do, the donor cells are not going to survive in the patient. Okay, that's the main thing that we're looking there. With the direct antiglobulin test, you can see on this slide that they can be used for quite a number of things. And we know from hematology too that anytime we saw spherocytes in a blood sample, the first thing we would think about is whether or not we have got a hemolytic component to the anemia, if in fact there is an anemia there. And we also know that there are other causes of spherocytosis, so we can have non-immune and immune causes of spherocytosis, and the direct antiglobulin test is such an easy, quick test to do that it can help us to determine whether or not we have an immune cause. So they are often used when we have anemia, so we're trying to work out whether it's immune or non-immune, but also very importantly, it's used to investigate whether or not we have had a transfusion reaction, particularly what we call a delayed transfusion reaction, which is due to our extravascular hemolysis, our IgG extravascular hemolysis, and also investigations of jaundice in the newborn. So we know that jaundice in the newborn, particularly those of you who have done clinical biochemistry, you know that there are a few other enzyme causes of jaundice in the newborn, but for us in transfusion, the one that we're mainly interested in is hemolytic disease of the fetus and newborn. So we use the direct antiglobulin test to give us an indication of whether or not that has occurred. So that's a really good question, Jordan. Would an ABO incompatibility resolve in neonatal, it's resulting in neonatal jaundice have a positive DAT? It can, because the reason that we get that neonatal jaundice in a patient like that, so if you have got a mother who is a group O and a baby who is group A and has inherited that group A from their father, then you can actually get ABO, hemolytic disease of the newborn. It tends not to be anywhere near as severe as the other forms of hemolytic disease of the fetus newborn, but you can get it. And the reason for that is because there is a small fraction of IgG with our ABO antibodies. So we have the majority is IgM, but we can have a small fraction that's IgG as well. And so that's what's causing it. And group O mums and group A babies are when it most commonly occurs. So that's a really great question. So you can get a positive DAT there. But we will be talking a lot more about that later on in the semester. Okay. So here is a rather severe case of spherocytosis and isocytosis, polychromasia, nucleated red cells with a clinical history of anemia. In fact, that's almost a leukoerythroblastic picture that we're seeing there. But of course, those spherocytes, if we see those on a blood film, that's an indicator that we need to do a DAT. And down the bottom here, what have we got down here? It's not so much rouleau, it's agglutination. Yep, so rouleau, you tend to have stacks of cells. So you'll have them sort of in a column or in a tower, whereas with agglutination, you tend to get these clumps, just as we see here. These are just clumps of cells that have been clumped together from antibody-antigen interactions. Yep, so anytime you see that sort of thing in the blood film, it's particularly common in people who have glandular fever, because glandular fever, you can create or you stimulate the production of an autoantibody, and we'll learn about that when we talk about our different blood groups next week. So that agglutination can tell you that you may have a cold agglutinin that is there, and you can detect that with the DAT as well. With these, the strength of reaction is quite weak. So again, your shaking technique with your tube is very, very important. No, it's not really a mixed field reaction, Amelia. It's different. You'll know a mixed field reaction when you see it. It's quite different to weak agglutination, and that's really all I can say at the moment. You should be experiencing mixed field reactions in one of your weeks of transfusion. Okay, so when we're looking at our direct antiglobulin test, the first thing we use is a polyspecific reagent in the first instance. So remember that polyspecific reagent, the poly and the specificity means the target, what it's going to react with, and the poly means many. So this means that this reagent will react with both C3d and IgG. And so that will tell us in the first instance whether or not we have a reaction there. And then if that reaction is positive, then we use our monospecific reagents. Okay, so we have a Coombs reagent that is specific for IgG only, and a Coombs reagent which is specific for C3d only. Okay, and so that will help us to determine what sort of a reaction we have. And these are particularly important when we're looking at our autoimmune hemolytic anemias. But they can also be quite useful when we, usually the query transfusion reactions that we get in the lab are delayed transfusion reactions, and they are IgG in nature if there is an immune component to it. The most common transfusion reaction that we get is actually a temperature one, so when temperature goes up, and so that doesn't have an immune basis to it. But we'll talk more about those in the week where we talk about the poorer things, the side effects of transfusion. So CHD is cold hemagglutinin disease, and PCH is paroxysmal cold hemoglobinuria. And you learned about both of these things in Hem2. So I suggest that you go back and have a look at that. The lecture in Hem2 when we talked about the immune basis of hemolytic anemias. Okay. So what are we up to? Right. So this is what you do when you do a direct antiglobulin test. You do a screen to see if there is any positivity there first, and then you try and zero in on whether it's likely to be IgG based using your anti-IgG reagent, or whether it's likely to be IgM based, or complement only based using your anti-C3d, because particularly with our drug-induced hemolytic anemia, sometimes we can have that complement being involved without the antibodies. Okay, so learning checkpoint 4, once again, just have a think about this. I'll give you 20 seconds to think. Which of the following will cause a positive indirect antiglobulin test? So maternal antibody coating fetal red cells, patient antibody coating patient red cells, patient antibody coating reagent red cells, recipient antibody coating transfused red cells. So why do you think D, Kiani? Good thought. If you have a look at the question, they are actually, so this is, think about where these things are happening. I guess that's probably the easiest way to work out whether or not it's a direct antiglobulin test or an indirect antiglobulin test. Is this happening within somebody, or is this happening in a test tube? Which one of those is not happening in the human body? So maternal antibody coating fetal red cells, that's happening in the fetus, isn't it? Patient antibody coating patient red cells, that's happening in the patient. Patient antibody coating reagent red cells is happening in a test tube. Yeah? Recipient antibody coating transfused red cells, so that's happening in the recipient. Yep, so C is the only one that is not happening in a body. Okay, so C is the one that's happening in a test tube. C is the indirect antiglobulin test. So just excuse me while I find my way back to the screens. Yep, so IAT occurs in vitro. Yes, Kiani, yes it does. Okay, so we are pre-sensitizing the red cells in vitro before we then wash the cells and add our Kuhn's reagent. Yep. So in the direct antiglobulin test, the cells have already been sensitized within a person in vivo. Okay. All right, specific AHG reagent contains A, anti-IgG, anti-IgM, B, anti-IgG, anti-C3D, or C, anti-C3b and anti-C3D. Yeah, so the polyspecific AHG reagent that we use contains anti-IgG and anti-C3D. Okay, so A is all about antibody and C is all about complement. Although when you look at them, I guess, you know, in theory, they're not exactly the same, but B is the one where we've got an antibody component and a complement component. And so that would be considered the most polyspecific, put it away. Okay, so this is just a really nice, again, a lovely flow chart of when, where and why to use the various reagents when performing a direct antiglobulin test. And I have put the URL on the week 3 discussion board for you so that you can have a look at that in your own time. It does take about 20 minutes though, so it's for those of you who are really interested, I guess, and it may just help you to understand a little bit more about the direct antiglobulin test. So in summary, the antiglobulin test is used to detect red cells sensitized by IgG alloantibodies, IgG autoantibodies, and complement components. The DAT detects in vivo sensitization, the IAT detects in vitro sensitization, and there are multiple sources of error that can be introduced into the technique. So you need to know that technique inside out and back to front, and you also need to know factors that influence it and the sources of error that can affect it. There's some review questions for you. This is a really, really important lecture and contains a lot of really, really important information that you have to know, okay? Because if you understand about the Coombs test, about the IAT and the DAT, every time you go into your prac, you will understand why you are doing what you're doing when you are screening your patient for antibodies or when you are performing a direct antiglobulin test. Okay, so thank you very much for your attendance, and that's the end of the lecture. Has anybody got any questions? No? Okay. Well, thanks very much. And I'll talk to you. I'll stop the recording now.